Welcome, dear students. Today we have to deal with the quality seeds. We have already discussed about the seed. Now, what is quality seed to which you can say that it is a quality seed? Because it is very important to provide quality seed to the farmers for assuring good crop yield. So in seed technology, this is very important to understand that what do you mean by quality seed. So here you can see that seeds with following characters. What are those characters? It must be having high germination percentage. It must be having high vigor. It must be having high purity. What is that purity? Purity, the genetic purity as well as the physical purity and the sound health. If your seed is having these qualities, means this is a good quality seed. This is a quality seed, you can say. So now let us discuss one by one how can you say that this is having the high germination. According to Seed Act, the minimum seed standard have been formulated for each and every crop, so that we must know about the minimum seed standard of different crops, that how much germination percentage, how much moisture it should be having, how much genetic purity, everything have been described in that. So that will be discussed later in other chapters. Today we have to concentrate about these things. If you see this high germination and high vigor, this you can say these are the physiological traits. Your variety, your seeds should have high germination. Then only, of course, the germination can be tested in the laboratory. But you see, by providing proper environment to the seed, in any time you can test the germination percentage of a seed lot by providing proper environment to the seed in your seed laboratory, and you can judge and you can test it. But if you talk about the high vigor, the establishment of, you can say, the germination as well as the establishment of the seedlings. Here germination cannot say that the normal establishment, cannot explain about the normal establishment of the seedling. So vigor means once you have planted the seed in the field, how much standability, how much establishment of the seedlings, whether it is normal or not. Then complete physiological traits will be seen. So whether your seed is having high germination, and if it is not having normal seedling establishment in the field, so it is not good quality seed. So the physiological traits should also be met of your seed lot. Then come to high purity. In high purity, you must have to see, the first is high genetic purity. Your seed lot must be genetically having the genetically high purity means there should not be any seed of other varieties of the same crop. This is the genetic purity of the seed. The seed should not have the seed of other varieties of the same crop. That is the genetic purity that has to be tested, and that has to be reported, that what is the purity of the seed lot. So it must have high genetic purity, otherwise it will be rejected as quality seed. Then come to physical purity. We have seen the physiological parameter, we have seen the genetic parameter. Now the next is physical parameter. The physical high physical purity must be there. What do you mean by this high physical purity? So it means the seed lot should not have seed of any other crop. Otherwise, that will be non-seed. So the seed lot should not have the seed of other crops. It should not have the inert matter. It should not have any debris or staples of the crop. It should not have stones, sands. It should not have the seed of your weeds. It should not have the broken seeds. All these things are coming your physical purity. So your seed lot must have high purity, that is high genetic purity, that is high physical purity as well. Now come to sound health. If your seed lot is meeting this quality, sound health, then only the complete seed lot is said this is a quality seed. So what do you mean by sound health? It means the seed lot must be free from your seed-borne diseases. It must be free from the infestations of the insect pests. It should be free from the insect damages. Then only you can say that your seed lot is healthy. Having the sound health. So this is all about the quality seed. Now you come to the role of quality seed. What do we mean by the role of quality seed? You see that one by one, it is a carrier of new technologies. Do you accept it? You have to accept it. Because whatever technology, so many technologies in agriculture you are having, if the main thing is not coming to the field, all technologies are useless. I mean to say that it is a carrier of technologies. Among all the technologies, varieties are the main technology, you can say. The new varieties are coming means you are providing new technology. And new varieties means new technologies, new varieties without seed. You cannot think about the varieties. So you are coming with new technologies in the form of new varieties, new yielding varieties, in the shape of good quality seed. So the seed is the quality seed is the carrier of new technologies. And you see that for each and every varieties, we have to standardize all the package of practices, all the technologies, For this variety, how much spacing is required? This is very important. We don't do this. We must do it. How much spacing is required for this particular technology, this particular variety, this new technology? Or this new technology, other new technologies has to be have to be evolved. That how much fertilizer requirement is there for this particular variety? What is the optimum sowing time for this particular variety? Each and every, what should be the spacing? Each and everything must be standardized in the form of new technology for that particular technology. So we can say it plays this quality seed plays good role as a carrier of new technologies to the farming community. Then come to next, it is a basic tool for secure food supply, as you know very well, that whatever green revolution you have achieved in 70s, you see what was your population in 1950s, what was the production, total food grain production, and after that what today you are producing. So see all those differences, particularly if you see the green revolution you have achieved because of the good varieties, because of the dwarfing genes have been introduced in wheat and rice, which became fertilizer responsive, losing tolerant, and because of that potential, you are now in the stage of export. You are exporting a lot, particularly wheat, and if you talk about wheat and rice, which was being imported a lot earlier before your grain revolution. So how it has been done? It has been done by the varieties in the form of good quality seed. You could achieve, you can secure the food supply. So similarly, this is a continuous process. The breeding is a continuous process to meet the challenges of ever-growing population, to feed them. So seed is the main input, you can say. It has to play its role for food security. Next, principal mean to secure crop yield in less favorable areas. What do you mean by this? You see, in many areas, if a particular crop is not being grown because it is not favorable to the area, that particular area. So if you are developing such variety which can grow well in those areas even where it was not being earlier grown, like you see in Punjab, now rice is being exported. Basmati rice is being exported from Punjab. Wheat. So wherever possible, wherever less favorable areas are there, if you are willing to grow a particular crop, if you are developing such varieties, and if you are providing quality seed to those areas, you can assure the crop yield in those areas also. So principal mean to secure crop yield in less favorable areas, this is one, that in those areas this can be grown. And other things you can see that less favorable areas means, if sometimes you see in those areas where the occurrence of flood is more recurrent, or in those areas where occurrence of drought is more recurrent. So if you are having the varieties for submergence tolerance like we are now having Swarna Sub 1, thousands and thousand acres rice crop is protected because of the single gene, Sub 1 gene, submergence tolerant gene. So you are having Swarna Sub 1. Likewise, there are drought tolerant genes DTY, drought tolerant yield QTLs. DTY gene 1.1, QTL 1.1, 1.2, 2.1, 2.3 means the QTL available on chromosome number 1, 1.1, chromosome number 2, 2.1, 2.2. Likewise, the submergence tolerant QTL available on chromosome number 1, you are calling it sub 1. So, if you are providing the seed to the farmers of this type of varieties, seed of these varieties, where floods are coming, submergence tolerant seeds you are providing, so they are protected with the loss of flood. So, this is the meaning of principal mean to secure crop yield in less favorable areas. Medium for rapid rehabilitation of agriculture in case of natural disaster if you are having. If say for example you have just transplanted the rice, or if you have just sown the wheat or any other crop, so lot of flood has come for a few days, everything lost in whole area if you say. Likewise, if you are in wheat or in other crops where because of the drought you are not having irrigation potential, so everything you have lost. So what you are going to do with that? So if you are having the reserve of seed, so this is the by establishing national seed reserve stock. If you are having the seed reserve stock, then in these bad days, you can immediately provide the reserve stocks of seeds to the farming community, that you don't have any seed to grow, so everything will be finished. So take these reserve seeds and grow it again, and so that you can have the crop production of your areas. So this is total about the quality seed and role of quality seed in agriculture. Thank you very much.